Welcome everybody to a Striatech hosted journal club. My name is Thomas Münch and I am one of the founders of Striatech. In our journal club series, different scientists present their projects and new data, highlighting the use and the application of our OptoDrum device. Today I'm very happy and pleased to welcome Elmar Hulliger as our speaker. Elmar worked at the University of Bern in the lab of Sonja Klein-Logel. He will talk about restoring vision, optogenetic gene therapy targeted at human on-bipolar cells. Before Elmar's talk, I will give a short introduction. Please let me highlight that throughout this event, you can ask questions, and we will have time to answer and discuss your questions at the end. A quick word about Striatech. Striatech is a dynamic biotech company from Tübingen in Germany. We specialize in the development of neuroscience testing tools, in particular for vision research. The Striatech founders are all experienced neuroscientists in the field of vision research. Progressive death of photoreceptors leads to blindness, for example, in diseases like retinitis pigmentosa. One very elegant way of potentially treating blindness is to make the retina light sensitive again. Since the advent of optogenetics about 15 years ago, there was tremendous progress in this field. Optogenetics refers to a method, namely to take the gene of a light sensitive protein, to introduce that gene to the target tissue and to express it there. From the beginning, there were three main questions. First, which optogene should be used? Initial studies focused on channel rhodopsin, a light-sensitive ion channel first discovered in algae. Second, which cells should be targeted in the retina to best recover vision? And third, how to get the genetic construct in there with efficient vectors. Finally, one problem one should not lose sight of is the intended application at the end, the human patient. Development of the methodology is usually done in tissue culture and in animal models, but at the end, it has to work in humans. In today's talk, Elmar Hulliger will share his exciting journey that touches on these three last topics. He will tell us about the development of a novel promoter that very efficiently targets on-bipolar cells and drives strong expression there, that is small enough to fit in adeno-associated virus, or AAV, which is the favorite virus for clinical applications, and that works efficiently not only in animal models, but also in the human retina. Before we hear from Elmar, I want to briefly touch on the first point here, optogenetic constructs. As I have mentioned, the first proof of concept studies introduced channel rhodopsin into the retina. One disadvantage of channel rhodopsin has been that it is not very light sensitive. It took enormously bright light stimulation to trigger retinal responses. In fact, so bright that a patient would hardly ever encounter such light intensities. There has been a lot of effort, quite successfully, to improve the light sensitivity of channel rhodopsins, but quite interestingly, this initial problem triggered other approaches as well. One of these approaches has been to use the native opsins, which are normally expressed in the retina anyway, and ectopically express them in the surviving retinal neurons. And it works quite well indeed. And this is also the approach taken by Elmar and his colleagues in the study we hear about today. Elmar will also tell us about his experiments where he evaluated the treatment success on the level of animal behavior. In general, when you do biomedical experiments in animal models, one of the challenge always is to properly quantify the disease and the effectiveness of your treatment. In vision research, you want to be able to quantify the visual abilities of your animals. This is where Striatech's OptoDrum can help, and we will see this in Elmar's talk. The OptoDrum takes advantage of the optomotor reflex. The optomotor reflex is a visual reflex that makes the animal follow global motion in the environment. Experimentally, you can evoke the optomotor reflex with a rotating stripe pattern surrounding the animal, as seen here. Elmar could show with our OptoDrum that his new treatment approach restores visual behavior in these animals. This tells us not only that the treatment worked, but it also tells us that the treated animals could see at normal light levels, that they could see stimuli on a computer monitor, so light levels that are encountered in everyday life. With this, I am very happy and pleased to welcome Elmar Hulliger as our speaker. Elmar attained his master's degree in biotechnology from the ETH in Zurich. Interested in applied research, he subsequently joined Professor Dr. Sonja Kleinlogel's lab of translational optogenetics at the University of Bern for his PhD. There, he established a translational model of post-mortem human retina, retinal explant culturing, and designed and evaluated synthetic promoters intended for optogenetic gene therapy. And after graduating, he continued his research in molecular biology and directed a preclinical trial of next-generation optogenetic gene therapy. Before you start, Elmar, let me please remind the audience that you will be able to ask questions throughout the event and also to give a thumbs up to other questions. A quick word about asking questions, because the interface can unfortunately be a bit confusing. When you want to ask a question, the cursor by default is in the name field. Giving your name is optional, but you have to click into the question field before you start typing your question. Otherwise, you will type your question into your name field. The Q&A panel on the right will stay available for a couple of days after the event. So if you are currently watching a recording of the event and Q&A is still available, please make sure to enter your question and your name so that we can back to you with the answer. We are very much looking forward to your questions, and Elmar, the stage is yours. Please go ahead with your presentation.
Okay, welcome everyone to my part of the presentation. Thank you, Thomas, for the introduction. So I'll be presenting the paper we brought out last year, named Empowering Retinal Gene Therapy with a Specific Promoter for Human Rod and Cone On-Bipolar Cells. So let me start by telling you about on-bipolar cells. On the top left, you see a schematic of a human eye. On the right, a simplified schematic of the human retina. So light is received by photoreceptors in the outer nuclear layer, cones and rods, and relate to bipolar cells in the inner nuclear layer. So bipolar cells are the first interneurons in the retina receiving a direct light input from the photoreceptors. There's broadly two classes of on-bipolar cells, the off-bipolar cells and the on-bipolar cells. On-bipolar cells are the ones responding to increments in light. In the on-bipolar cell class, there's two classes, which are rod bipolar cells receiving their input from rod cells, and cone on-bipolar cells receiving their input from cone photoreceptors. So then bipolar cells relay the signal from the photoreceptors to the ganglion cells. Important to note is the macular fovea region of the human eye. The fovea is cone dominated, so cone on bipolar cells relay the signal there. It's responsible for central vision and also has the highest visual acuity. Therefore, it's a very important target for human gene therapy. Okay, so in retinal degeneration, especially in late stage, photoreceptors are gone. However, the inner nuclear layer is intact for extended periods of time, which gives us an opportunity to introduce light-sensitive proteins to the surviving cells. So optogenetic tools targeted for on-bipolar cells are opsins and their G-protein coupled receptors, which are able to integrate into the signaling of the on-bipolar cells. We used OPN1 middle wavelength in this work to render on-bipolar cells into kind of replacement photoreceptors. So they are able to detect light and send their signal to the ganglion cells. Because they take advantage of the signaling cascade on bipolar cells, so the amplification through second messengers, it makes them highly light sensitive compared to traditional tools like channel rhodopsin. A further advantage of targeting on bipolar cells is that since they are the first interneurons of the retina and the visual signaling cascade, one can reactivate the processing of the internuclear layer and therefore potentially achieve very high quality vision restoration. So yes, we in our lab were mostly focused on optogenetic gene therapy. However, I'd like to say that not only for optogenetic gene therapy, but also for supplementation therapy, also known as replacement therapy, on bipolar cells are interesting targets. What you see here are proteins in on bipolar cells, which when mutated, so the proteins denoted with red stars, when mutated can lead to congenital stationary night blindness. So if one could insert a healthy copy of a mutated gene, one could alleviate the night blindness. Important to note is this receptor here, it's the metabotropic glutamate receptor 6, which actually receives the input from the photoreceptors. And its gene name is GRM6, which I will be using quite often in this talk. So just to make it clear that I'll be talking mostly about the promoter of GRM6 and sometimes about the gene GRM6. Also important to note is that this protein is expressed in on-bipolar cells very specifically. Okay, so if on-bipolar cells are such interesting targets, why exactly is there no gene therapy which targets them at the moment? So what was missing? You heard from Thomas that gene therapies have 3 parts. So the first part is the vessel that brings the therapy to the right cells. You heard that the adeno-associated virus AAV is the current vector of choice because it's non-pathogenic and also small with really good tissue penetration. Basically, the only drawback of it is that it has a limited packaging capacity of approximately 4.7 kilobases. This means we cannot use 10 kilobase long endogenous promoters to do the job, but have to resort to synthetic shorter promoters. The second part of a gene therapy is the optogenetic tool or the replacement gene. Of course, the replacement gene is easily come by. And as you heard, we have a very effective optogenetic tool, which are being further refined all the time. So this part is also okay, not a problem. So the third part is the promoter, and it turns out that up till recently, no promoter was up to the task of targeting on bipolar cells in human gene therapy. Of course, since on bipolar cells are promising targets, work has been done on this. One very important effort was produced by Kimedal in 2008. He looked at the endogenous murine GRM6 promoter, which you see over here. So it's around 10 kilobases long, and then performed deletion studies to see what elements exactly produce the results, produce expression in on bipolar cells. So generally, the idea is when designing a promoter specific for a cell is you want to mimic an endogenous promoter, but with less space. So what they found, and they used always kind of a combination of an enhancer region and a proximal promoter or basal promoter element. So what they found is that there is a 200 base pair, really small enhancer element that when present in this enhancer promoter combinations led to expression in on bipolar cells, which you see here on the left. while when it was missing, so in all the other cases, no expression was present. This means that this 200 base pair enhancer was both necessary and sufficient to drive to activate expression in on-bipolar cells. I combined it with a SV40 basal promoter, so a viral promoter, and saw that they got expression in on-bipolar cells of rat and mice. They also aligned this 200 base pair enhancer sequence of the mouse with the human genome and found a 183 base pair sequence there in the human GM6 promoter, which correspond to this 200 base pair enhancer. Oh, then in 2014, Cronin et al. Improved on this promoter. So you can see here basically the structure of these promoters is always a promoter element, so core proximal promoter element, and then an enhancer. And Cronin et al. Used a quadruple repeat of this 200 base pair enhancer combined with the SV40 basal promoter called 4x GM6 SV40. And they saw that this led to higher expression. However, we in the Klein local group tested this in RD1 mice and RD10 mice. So RD1 is a harsh retinal degeneration model with a fast onset of degeneration, while RD10 is somewhat milder. And we found the following. So first I'll explain the staining quickly. So here in the bottom left, you see in red, rod bipolar cells are marked by PKC alpha. And on the right, all on-bipolar cells, so including the cone on-bipolar cells, are marked with GO alpha. And the reporter protein driven by 4x GM6 SV40 promoter you see in green. So you can see there is expression in on-bipolar cells indeed. However, there is also a lot of off-target expression in the internuclear layer as well as the ganglion cell layer. So this 4x GM6 promoter was actually not specific in the RD10 retina. But much more important, in the top you see RD1 experiments, actually at age P53, so about 7.5 weeks old. It's pretty young. And again, on-bipolar cells are labeled by in red by gamma Y13, G gamma 13. And you can see that there's barely any expression in on-bipolar cells. So there's one over here, and one weakly expressing over here, but the majority of expression is in amacrine cells of the internuclear layer as well as in ganglion cells. So of course, this is a no-go for a promoter intended for human gene therapy. The promoter has to be active in the target cells of the diseased tissue, and therefore it was necessary to find a promoter that was able to do that. Maybe as a quick overview of all the requirements for a promoter intended for human optogenetic gene therapy targeting on-bipolar cells. It has to be short because it has to be fit next to the transgene in the AAV. It has to be specific for on-bipolar cells, including cone on-bipolar cells, because these populate the macula, which I told you is the most important target for human gene therapy. And the 4x GM6 promoter is small enough, but it was not specific for on-bipolar cells, at least not in the RD1 and RD10 mouse models. Should be functional in the disease state, of course. It should be functional in the intended target tissue. So I put the tick here in parentheses because we saw some expression with 4x GM6 SV40, but it was unspecific and also pretty weak. And it optimally consists of human DNA to optimally be compatible with the human transcription machinery, and also to not elicit any immune responses from, let's say, for example, viral sequences. So you see that 4x GM6 SV40 was not fit for the job, and it was time to look for a new promoter. We want to also use GM6, or the promoter of GM6, of course the human variant, for promoter design, because it has this advantage of being highly specific for on-bipolar cells. And also we knew from Kim et al. A functional enhancer region, which is also valuable information. However, we were very worried from our experiments in the RD1 retina that this 200 base pair enhancer is actually, its function is downregulated in the RD1 retina, because if this would be the case, it would not make sense to base or promoter design on the human GM6 gene. However, it could also be the case that it was because of the SV40 basal promoter or interaction between those two. So our first step was to check whether GM6 is still expressed in the RD1 retina, because if it was, this would indicate that the enhancer function was still okay, but the problem would lie with the SV40 basal promoter. So, and if GM6 is still expressed in the RD1 retina, we could be base or promoter design on the human promoter of GM6 and test the promoters in relevant experimental models. So our models were post-mortem human retinal explants. This is a translational model since it's the actual target tissue, and therefore a very, very good model for this kind of experiment. And the other model we used is the RD1 mouse model. And the thinking was that if we manage to get expression in on-bipolar cells of this harsh degeneration model, it would also work in other less harsh degeneration models. Okay, so step one, is GM6 still expressed in the RD1 retina? For this, we did real-time qPCR experiments and compared the values in RD1 at different time points with the values in wild type. What we got was this. So in the black dots are the actually measured values relative to the wild type value, and the empty rhomboids are the values corrected for the loss of the outer nuclear layer, which of course has an impact on relative amounts of mRNA expressed in cells of an inner nuclear layer. So what we saw is that between P21, which is this one, and P28, which is this one, there was indeed a downregulation of GM6. However, after this time point, the downregulation slowed down, and the expression of GM6 remained stable for quite a long time. So up to very late, very old mice. Yeah. Also important to note is that if you remember, I said we did the RD1 experiments with 4x GM6 at about 7.5 weeks. So around this time point, and at this time point, the expression of GM6 was still at 60%. So we felt confident that the problem with 4x GM6 was more likely to be with the SV40 basal promoter than with the actual GM6 enhancer sequences. Therefore, we went for it and used the human GM6 promoter as a template to design new promoters. Maybe a bit on the strategy. We decided to not rely on bioinformatic prediction of transcription factor binding sites, because while bioinformatics is a powerful tool, certainly, and very useful for large-scale approaches, it still produces many false positives and false negatives. And in our case, like serial testing of promoters was not really possible because it takes a lot of work, including packaging viruses and etc. So we wanted a more targeted approach and decided to rely on annotated and experimentally confirmed data, which mostly means ChIP-seq data as well as cross-species conservation. Also, usually little is known a priori about how a promoter exactly functions. There's many parameters that influence promoter function. So in order to kind of preserve the functionality of the original promoter, we wanted to change as little as possible. So no wild switching around of transcription sorry, of transcription factor binding sites. So we basically wanted to cut and paste as little as possible. Okay, so this is the human GM6 promoter. On the right, you see the start of the actual gene. And you see that the promoter is over 10 kilobases long. And we focused on this enhancer region, which contained 183 base pair sequence identified by Kim et al. Corresponding to the 200 base pair enhancer, which showed pretty nice results in wild type mice. And we wanted to use a proximal promoter also of GM6 located at the transcription start site and translation start site. So first for the enhancer, so now this is a blow up of this spot here. We did not only align the 200 base pair murine enhancer, but a whole stretch, and found that actually the conservation between human and mouse was longer, which is the dark area here. It's around 310 base pairs. And I wanted to include this whole stretch in my enhancer elements. So basically I chose one version where this conserved region is encompassed, and one version which extends further upstream to also include these chipset peaks here, as well as the DNase cluster here, which is indicative of regulatory active regions. So these two were my enhancer elements, which I wanted to try out. And similarly, for the proximal promoter, I also chose two versions. So now we're here at the transcription start site, translation start site. And also here we found the conservation between the mouse and the human genome, which was, I think, 167 base pairs long, which I wanted included in the proximal promoter versions. And of course, the transcription start site has to be in there. And I chose one version extending further upstream to include this ERG peak, as well as the acetylation mark peak here, which is also indicative of regulatory active regions. And in my second version of the proximal promoter, I extended further downstream up to the translation start site, including this chipset peaks here. So basically, I had four elements which I wanted to combine in enhancer proximal promoter construct, which means I could produce four constructs. I chose to produce three, so I cloned those. And depending on the results, I still could have cloned the largest version with the long promoter and the long enhancer. The names in the paper are a bit of a mouthful, so for this presentation, I'll just call them by their length. So promoter short, promoter medium, and promoter long. As I already mentioned, we wanted to test in RD1 experiments and in human retinal explants. So for the RD1 mouse experiments, we used AAV with this expression cassette. So promoters driving an optogenetic tool, as well as a fluorescent reporter protein. AAVs have an onset of expression of around 4 weeks. We injected intravitreally and then evaluated with behavioral experiments and immunohistochemistry. For the human retinal explants, we could not use AAV because single-stranded AAV, because at the time we were not able to hold the explants in culture for 4 weeks. So we used self-complementary AAVs. These have the advantage that they have a faster onset of transgene expression. So at day 7, we could see fluorescence of the reporter gene, but they have only about half the packaging capacity of normal AAV, so even less. But still, yeah, we could fit the reporter protein in there, and and looked at expression after 7 days of culturing. Maybe a bit more in detail about these human retinal explants. We got the donor eyes from the university hospital, which is near the institute. Usually we got them around 2 to 16 hours after death. Eyes that were over 24 hours in age usually did not give good experiments, so of course the fresher the better. I took explants normally from the mid-periphery, roughly denoted here with this red area. You can also see the macula denoted with the blue area. Punched out 8 millimeter diameter explants, put them into hanging cell culture inserts with the ganglion cell side up. And then on the day of explanting, I added 3 microliters of virus to transduce the explant and processed the tissue at day 7 of culturing for immunohistochemistry. And what we got out looked something like this. So we were really happy about these results. So this is for promoter long. And you can see that most of all on-bipolar cells shown here are expressed in the reporter gene, which means it has a nice expression efficacy, and also that basically only on-bipolar cells are expressing the transgene. You see here in the ganglion cell layer one cell that weakly expresses the reporter gene as well, but it has really high specificity for on-bipolar cells. For promoter short, we saw almost as nice of results. However, with promoter medium, we found a problem, which was that it only expressed extremely weakly. We quantified this by quantifying the fluorescence intensity in non-bipolar cells, and which we normalized to the background fluorescence in non-expressing cells. And you can see that promoter medium actually is barely above background, while promoter long and promoter short showed nice and clear expression. So we decided to drop promoter medium. You can see it's the only one with this 566 base pair promoter, and continue only with the two other constructs. So what you did not see in the image before is the difference between cone on-bipolar cells and rod bipolar cells. Here I did triple labeling, and to make it a bit clearer, I split it into two parts. So on top, both G-alpha-O and PKC-alpha are in red, but in the bottom I changed the channel color of G-alpha-O to cyan. And here you can see that cells that are only labeled by G-alpha-O are cone on-bipolar cells, and cells that are labeled by both PKC-alpha and G-alpha-O are rod bipolar cells. And you can see that all these cells that I indicated in the top picture are actually on-bipolar cells, so only labeled by G-alpha-O. We evaluated the efficacy, so how many of all cone on-bipolar cells were expressing, and this was, for this example, 56%, but for rods it was slightly higher. However, I wanted to have a kind of a measure which told me what was the preference between for either cone or rod bipolar cells of my promoter, and which also was not dependent on how well the experiment went. So if there was little expression, basically I wanted a number that I could use to compare with different experiments. So this was the cone on bipolar cell preference, and basically it is the expression efficacy in cone on bipolar cells normalized by the sum of the expression efficacies of cone on bipolar cells and rod bipolar cells. So this cone on bipolar cell preference basically tells me that if it's 50%, it means that my promoter does not preferentially express in either cone or rod bipolar cell. And of course, if it's 100% rod bipolar cells, it means that it will only express in rod bipolar cells. And also it means that if there were an equal amount of rod and cone on bipolar cells in a given area, which most often is not the case, one would expect with a 50% cone on bipolar preference to see the equal amount of expressing cells of both rod and cone on bipolar type. So in this example, the cone on bipolar cell preference was 45%, and respectively the rod bipolar cell preference was 55%. However, we quantified that properly, and we saw that actually promoter long had almost equal preference for cone and rod on bipolar cells, while promoter short had a lower preference for cone on bipolar cells. So clearly promoter long was our best candidate from these three promoters. And we continued evaluating promoter long. Another way to see if this promoter really works in cone on bipolar cells, and also a really nice proof of concept experiment, is transducing the macula. On the top left, you see the schematic of the macula with indications of where the pictures are. And we did that, and you can see that in the macula, where basically, especially in B, D, and E, basically there are no rod bipolar cells present, but only cone on bipolar cells. We got also a nice expression in those cells. So this was a nice proof of concept experiment, since most gene therapies are targeted at the macula. We also looked at spread of expression of promoter long. So there's a lot of information on this figure, so let me take you through it. In the top left, you see a flat mount of a human retina. In blue, you see the pipette marks are left while placing the virus on the explant. So this is a little bit of damage on the retina, and red, you see a blood vessel. Now, interestingly, if one would assume that that the virus accesses the inner nuclear layer by the sides of the explant or by damaged areas, and not actually by penetrating the inner limiting membrane, the ganglion cell layer, to reach the inner nuclear layer, one would expect to see expressions centered around these damaged areas. But you can see that also far away, there's really nice expression, which means that this virus actually readily penetrates through the inner limiting membrane to the inner nuclear layer. Yes, then in B and F, you see just different examples of different donors. Again, marked in red with G-alpha are bipolar cells. And you can see that we found expression efficacies of from 65% to 80%. Yes, and in the bottom is yet another way to check for expression in cone-on bipolar cells. And the point being that in the inner nuclear layers, rod bipolar cells are located in the outer part of the inner nuclear layer, while cone-on bipolar cells are more distributed and more in the middle and inner part of the inner nuclear layer. And what you see here is basically 3 depths in the inner nuclear layer. So this is close to the inner plexiform layer, so the inner part of the inner nuclear layer, where there's no rod bipolar cells. This is the middle of the inner nuclear layer, and this is the outer part of the inner nuclear layer where there's mostly rod bipolar cells. And you can see that irrespective of depth, there's about the same expression efficacy, which you can see here in the top right corner, which again is telling that promoter long has no preference over cone or rod bipolar cells over the other. So we also evaluated the on-bipolar cell specificity, of course, very important. We did this with 2 different capsids, one 7M8 and another 6Y12 mutation, just to see if also the capsid has a role in cell targeting. And we saw very similar results with both, which is almost 90% of on-bipolar cell specificity and very little off-target expression in other cells, so in the outer nuclear layer, inner nuclear layer, or ganglion cell layer. We also evaluated the expression strength via fluorescence in rod bipolar cells, cone-on bipolar cells, and compared them to the expression strength in cells in the outer nuclear layer, inner nuclear layer, and ganglion cell layer, and found that generally the expression on bipolar cells was stronger than the off-target expression in other cell types. So taking G and H together, we can say that promoter L produces very little off-target expression, so very little amount of cells which are not on bipolar cells are expressing the transgene, and in those cells, this transgene is weakly expressed compared to the expression in the outer nuclear layer. If you combine this with a cell-tailored tool that is tailored to work in on-bipolar cells, this makes it very unlikely to have negative side effects from off-target expression. Oh, we were very happy with the performance of promoter long in the human retina, but we still had to test how well promoter long fared in the RD1 retina. So as I said, we used AAV and we used OPN1 middle wavelength driven by promoter long, as well as a reporter protein. So this was the expression cassette. We injected 22-week-old, so pretty old RD1 mice intravitreally, and then used the Striatech OptoDrum virtual optomotor system to measure the optomotor reflex and determine visual acuity of these treated mice, comparing them to untreated mice and wild-type mice. And what we found was the following. So we tested at different ages, so up to 38 weeks of age, and we did not find any difference in those, significant difference in the values we saw there. And what we saw is that the treated RD1 mice had significantly higher visual acuity than the untreated litter mates, and even and the restoration of this the vision recovery was actually about 65% of the visual acuity of wild-type mice, which you see here on the left. So this was a very important experiment because it told us first that promoter long was strong enough to express at therapeutic levels, because you cannot see this from immunohistochemistry. So it was effective, and also that it expressed in enough cells to make a difference, so to really make the therapy work. So this was a very important proof of concept experiment, and we're really happy to see that. Also, we looked at the immunohistochemistry. So here on the left, you see a flat mount of a 41-week-old RD1 mouse. So actually one of the mice that went through the OptoDrum. And you can see that the expression is widespread and essentially pan-retinal. In the middle, you see the expression in a 26-week-old RD1 mouse. Keep in mind that with the 4x GM6 promoter at 7.5 weeks, we did basically saw no expression on bipolar cells, and this 26-week-old RD1 mouse shows really nice expression in on-bipolar cells. So this was also very good to see. We measured the on-bipolar cell preference and saw that with 60%, it was a bit lower than what we saw in the human. This might be because of the degeneration, but also maybe because of the difference between the human and the mouse retina. However, it was still specific for on-bipolar, well, a kind of specific for on-bipolar cells, and certainly enriched. And most importantly, it expressed enough in on-bipolar cells that the therapy was actually effective. So taken together, we saw that promoter long, so this is the actual name in the paper, promoter long is short enough for AAV use, because we designed it that way. We saw that it was specific for on-bipolar cells, including cone on-bipolar cells, so it had about 90% specificity in the human retina, and basically equal preference for cones and cone on-bipolar cells and rod bipolar cells. We saw that in the disease state of the RD1 mouse, it expressed at therapeutic levels. So we could have an effective recovery of vision. It was, of course, functional in the human retina with widespread on-bipolar cell specific expression, even in the macula. And because we designed it that way, it consists of human DNA. So the requirements that we set out to comply with were met. And yeah, I really hope to see clinical applications using this promoter in the future. So with this, I would like to come to the end and to my acknowledgments. So I thank my co-authors, Sonja Kleinlogen and Simon Hostetler. I also thank Volker Enzmann, through whom I had access to donor eyes. I thank Ivan Arsenjevic, who sent me actually the medium for human retinal explant culturing. I also thank Pascal Escher, who gave me tips for the real-time qPCR experiments. And from the lab, I thank especially Michael van Wyk, which did the mouse injection, Sabine Schneider and Lara Girot, which did most of the viral packaging, Michael Kanzig, who took care of the animal facility, and Nina Stocker, who helped out with immunohistochemistry. And of course, I also thank the rest of the Klein local group. So with this, I'm finished, and I thank you for your attention.
Thank you, Elmar. Thank you so much for the presentation. It was very interesting, and we've also had quite a few questions coming in. And if you don't mind, we'll dive right into these questions.
Sure.
Okay, let's see. I have to read this. So the question was if you ever considered using different carriers other than AAV, for example, nanoparticles, to avoid the size limit that AAV gives you?
Well, we did not. It is, of course, a different way to get around the problem. but we want to take advantage of the advantages of AAV, and therefore we went for a short promoter. Of course, in therapy, nanoparticles. I actually don't know what the size is you can load on nanoparticles, but I imagine it is pretty unlimited. So you could, in theory, use the endogenous GM6 promoter. So this would be a different way.
Okay. And another question was you went for GRM6. Did you try any other on-bipolar cell specific promoter?
Yes, I did. So initially, I looked at multiple candidates of gene promoters to base my design on. For that, I mostly relied on a paper from Siegert et al. From 2012, which basically is a map of which genes are expressed in which cells in the adult mouse retina. There, I looked at which genes were expressed in on-bipolar cells, but not in other cells. Found multiple candidates. Looked for corroborating evidence in the literature, and also for which of those genes already had promoters, short synthetic promoters made out of. For example, there's the Pleiades promoter project, which did produce quite a lot of promoters. I chose basically a few good candidates. I did the real-time qPCR in the RD1 and looked at the expression in RD1, and kind of putting together all these factors, I decided to try out 3 further promoters. So one was TNNT1 based, which actually was from the Pleiades project. Another one was TRNP1, which had really high expression in the RD1 retina, and the third one was TRPM1, which already also had kind of promoter used, but completely unrelated to the retina. I think it was in melanoma cells. So basically, yeah, I tested these 3 versions of promoters from different genes, and both TRNP1 and TNNT1 gave sporadic expression in all kinds of cell types, but not in on-bipolar cells. And TRPM1 gave expression on bipolar cells, but really unspecific. So the internuclear layer as well as the ganglion cell layer was full of expression. So yeah, it's actually pretty hard to hit on bipolar cells, and yeah, GM6 seems to do the trick.
Yeah, sounds like it. There are 2 questions that I guess go a little bit in the direction of safety studies. One question is if you know whether your promoter would drive expression in non-human primate retinas to be able to do safety studies in those.
Well, you don't know until you try, right? But I think it should be able to drive expression in non-human primate on bipolar cells, because if it does so in the human and in the mouse, kind of the non-human primate is somewhere in between. So I would totally expect it to work, yes.
Okay, and along that line, do you know if there is any expression in cells in CNS and other CNS neurons, not necessarily retina, but if you can get ectopic expression of GRM, natively the protein mGluR6, or your promoter in other CNS neurons?
Actually, I know that the GM6 is basically only expressed in on bipolar cells, but in the retina, and I'm not so sure where else GM6 is expressed in the central nervous system. I think so in what cell populations, but I would expect that in cells that it's not a given, but I think that in cells that express the mGluR6 in the central nervous system, most likely this promoter might actually also work. Yeah.
Mm-hmm. Then a very detailed question. So I don't know the answer to that, but the question is whether you checked the promoter described by Massé et al. From 2015.
No, no. I actually no, I don't know. I know about the promoter by Lou et al. From 2016, but the other one doesn't ring a bell, I think. So now I cannot ask how it worked and what it was based on. So sorry, no, I don't know.
So obviously you did not check,
No.
right? So then you can answer that question that you didn't check it. Another question was, I guess in the mouse experiments, whether you treated both eyes or whether you used one eye as mock injection control.
We injected both eyes. And also we analyzed all the eyes with immunohistochemistry. So no, but in one of these mice, it was clearly one eye had the much better expressions and the other had very little expression. So not always the experiments are a success. And even in that mouse, the visual acuity actually was not significantly different. So probably it compensated by just having one eye that really kind of worked. Yeah.
Mm-hmm. Okay. Do you know about the duration of expression post-injection?
Concerning possible downregulation after a long time of my promoter, I think. What was the oldest mice we looked at? I think we looked at mice of about a year. Was probably the latest we looked at. The problem is that, especially in the RD1 mouse model, the further you wait with experiments, the less nice it will look. For example, the labeling of on-bipolar cells is quite tricky. Sometimes it works nicely, so the G-alpha and PKC alpha labeling, and sometimes it doesn't work at all. So it's really difficult to evaluate them. But yeah, I think, I don't know exactly, but I think up to 1 year we still saw expression. Yeah.
Okay, in mouse. And a similar question in the explant culture. Of course, it's not very easy to keep these cultures alive for very long. So one question was how long you can take them alive to potentially see off-target expression in cells that just, where it just takes longer for the gene to express. Mm-hmm. And how, so in general, how long can these cultures be kept? And second, how long did you keep them?
I think nowadays there are a few groups that can keep them for very long indeed, so weeks and weeks and weeks. However, I basically, when I started, when I got to being able to keep them 1 week, I was very happy, and then I just did the experiments with self-complementary IRV. And towards the end of my PhD, we looked if we could extend this time, and I did a few experiments with 3 weeks, and actually they looked pretty nice. So I think at the point where I finished, I think we could keep them for about 3 weeks, 3 or more. I mean, we didn't check exactly, because of course it would be nice to actually use single-stranded AAV in the human retina, not only because it's a more proof concept experiment, but also because of the packaging capacity, and then you could do electrophysiology on the actual human tissue to see if it's also produces at therapeutic levels in the human retina, which I expect it does.
Mm-hmm. Yeah, a whole bunch of people asked if you had any other functional readouts other than the optomotor reflex, in particular ERG.
I did not, but I also was not the electrophysiologist. I think by now they had have a bunch of other readouts. So I know the lab I was part of, they do MEA experiments, and also, yeah, of course, ERG, and also visual cortex readings. So if you want to know more about this, I urge you to ask Sonja Kleinloge.
Okay. Since you injected both eyes, you mentioned, you can probably not answer the next question. The next question was whether there's contralateral AAV transmission. If you inject in one eye, that you actually see expression in the other eye.
Yeah, I cannot answer this.
Yeah. Uh-huh. So next question. First, I have to read it myself. Excuse me. Yes, whether you could see any toxic side effects of any way of your promoter. Probably difficult to say since this retina looks pretty harmed anyway.
Yeah, because it's hard to differentiate. So of course in the human retina, it was pretty a short culturing time, and so it would have to be in the mouse experiments, and there it was RD1. So you exactly know if it's just a degeneration or if it did additional harm. But in terms of the degeneration, I think that the therapy might even have a kind of protective effect by reactivating the internuclear layer and activating those cells. Since we didn't see any difference between, 26 weeks of age and 38 weeks of age, which we would expect to still, so we would expect the condition to still worsen, but we did not specifically quantify any such adverse side effects. Yeah.
Mm-hmm. Did you ever inject wild-type retina to see for potential deleterious effects?
We injected in wild-type mice as well, but primarily to look at basically the expression patterns and the effect it's also the positive effect in the case of RD1s, but no. So we did not really look at I don't know, inflammatory responses or something like that, and most negative effects we saw in injected mice was actually because of the anesthesia. So no, we did not.
Okay. Then there's a question if you ever considered doing multi-electrode array recordings with the human retina.
Yes, this is the job of my former colleague Jakub Kralik. So he's doing this right now, and I think, well, I don't know if he's doing it right now, but this is the goal. He's certainly doing it for mouse retina, and of course the goal is to use the human tissue as well for multi-electrode array recordings.
Mm-hmm. Yeah. If there were enough space in your AAV, do you think it would help to put your long promoter also in tandem behind each other?
So as in 4 times 3
the enhancer region, yeah. At least, right? The enhancer, double the enhancer region.
Yeah, I think one could maybe boost the expression. However, I mean, too much is also not optimal because it puts strain on the cell, and maybe too much expression would also increase off-target cells. One could also optimize different aspects of the promoter, maybe make it even shorter, cut away parts that may not have any function. However, all in all, I think it works as intended, and if it ain't broke, don't fix it. I would not really do it. Yeah, but I mean, one could do experiments and see what effect it has, but I don't think it's necessary.
Mm-hmm. Our time is almost up. Maybe one more question. So the question is, why is it enough to express the opsin there? Why do you not need all the other components of the visual cycle that are part of the visual transduction cascade normally in the photoreceptors?
Well, I'm not an expert on the opsins themselves, but I know that this opsin at least is able to integrate into the signaling cascade of the on-bipolar cell, so the GI pathway. And for a different optogenetic tool, OptoGenetic, for example, which is a chimera with a light-sensing part of melanopsin and signal transducing part of AmGLR6, it's maybe the most obvious because it actually for the interior of the cell, it looks exactly as a normal on-bipolar cell, only that the kind of AmGLR6 receptor is activated by light instead of glutamate.
Mm-hmm. Yeah. Okay. Elmar, thank you again so much. Thank you also to the audience who participated with a lot of interesting and good questions. For those questions that we did not have time to answer them, we will answer them in writing. Also the questions that come from people that look or watch a recording of the event, we'll answer them in writing, and we will post the questions and the answers on our website, on the Journal Club page for Elmar's Journal Club today. So thanks to everybody, Elmar, especially to you for participating, and have a good day, everybody. Thank you.
Bye.